Now, I would like to introduce my colleague, Mary McHugh, Professor of Greek, Latin, and Classical Studies, who will introduce our first speaker for the day. Mary?
Carl June is the Richard W. Vague Professor in Immunotherapy at the Abramson Cancer Center at the University of Pennsylvania. In 2017, the FDA approved the first gene therapy for the treatment of cancer, called Chimeric. It genetically modifies existing T cells in a patient's body, which enables them to target proteins on the surface of cancerous cells. In other words, it retrains healthy cells to hunt down and eliminate cancer cells. It was Dr. Carl June's lab at the University of Pennsylvania that developed Chimeric. June has been a leader in the development of immunotherapy, specifically chimeric antigen receptor therapy, usually referred to as CAR-T. This is a promising new form of therapy that offers a prospect of curing cancer using the immune system. The CAR-T therapy is currently being used to treat acute lymphoblastic and chronic lymphocytic leukemia, and trials have begun to use it for other types of cancer, including blood, pancreatic, and brain cancers. Today, Dr. June will discuss the promises and challenges faced by the evolving CAR-T cell industry.
Good morning, everyone. I'm recording this the morning after the first presidential debate. Quite an evening, I must say. I wish we could be together at Gustavus, but unfortunately we're virtual and I'm missing the interactions we would have had. My name is Carl June. I'm a professor at the University of Pennsylvania. My disclosures are listed here. The trials I have and will discuss have been supported by a variety of sources, including industry as well as the National Institute of Health.
Take one. Take one.
Is it hard for you to say those words? We're trying to cure cancer.
That's a really good question, and why it's hard to say we want to cure cancer. We do. And I think sometimes it's hard actually to think that you might actually succeed.
Patients that we're treating on this clinical trial have absolutely no other options left for them. These are patients who are unfortunately destined to die of their disease and in a fairly short amount of time.
Emily Whitehead, 554.
So Emma is incredibly matter-of-fact about all of this stuff. This was a child who had had her leukemia come back twice. The parents were looking for a miracle.
What we've learned how to do is train the immune system to recognize and then kill tumor cells.
It's a procedure where we collect their T cells, and they are infected with a virus that will genetically change them so that they will now see and react against their leukemia cells.
And we actually use the HIV virus to do that.
So you're taking the HIV virus and infecting healthy cells with it to help kill cancer?
Yes. The virus has been engineered so that it can't cause disease anymore. But it still retains the ability to reprogram the immune system so that it will now attack cancer cells. We call those modified immune cells serial killer cells. Each infused cell can kill more than 1,000 different tumor cells.
But the reality is the dramatic responses of cancer to new treatments are very unusual. We need to make it clear when we talk to a family that it may not work. Emma was given her T-cell treatment, and within a few days, she was very sick. She had breathing difficulties, she had blood pressure difficulties. We knew that she could not have gotten any sicker without actually dying.
But then a remarkable thing happened. The T cells were growing, they were starting to fight the cancer.
Within hours, Emma's fever disappeared.
It was like the calm after the storm. The clouds went away, and she woke up, and there was no leukemia. When that child survived, it was, of course, an amazing event.
So that video was recorded in 2012, and some follow-up since then. This is a Facebook posting from her father, Tom Whitehead, and you know, within a few days, that video went viral, and it had over 1 million views on YouTube. And I began to get emails from people with cancer or their loved ones who had cancer asking if we would infect them with HIV so that they would have CAR T cells. And this is what Tom Whitehead posted, that, you know, the public didn't understand that we were using an engineered form of HIV, a disabled virus that actually becomes a tool rather than the pathogen that kills us through AIDS. So that's one example of sometimes social media can be self-correcting. Emily then went in 2015 to the White House. President Obama had initiated with Vice President Biden something called the Precision Medicine Initiative, and it was in January of 2015. He invited her to the White House. At that time, she was 10. And after receiving line afterwards, he asked Emily and her father if there was anything he could do for them. And the father, quick thinking as always, Tom Whitehead, said, well, she's supposed to be in school today because it was a Monday. Can you give her an excuse? And Obama then wrote and gave him this White House stationery, a get out of jail free card. So our CAR T cell that we're discussing today stands for chimeric antigen receptor T cells, or CAR T cells, and it, you know, was initially named after the chimera, the Greek creature that is a fusion of three animals, a lion, a goat, and a serpent. But in reality, the reason it was called a chimeric T cell is it's a fusion of the cells in the body that normally make antibodies as well as kill infected cells, and those are T cells. So a little bit of Immunology 101. So normally a T cell, which evolved to kill viruses, it uses a very complex mechanism to activate the key killing properties of the T cells. And if this is the T cell here, it uses a very complex receptor that binds to different nucleic acid and protein complexes that are expressed on the surface of the virally infected cell, or it could be a tumor cell. Our CAR T cell uses a different approach, which is it has an antibody instead of the complex T cell receptor. It has an antibody on the surface, which is derived from a B cell, inserted into the T cell using the lentiviral vector technology, and then it can then kill the cell based on any protein that's selected on the surface of the target cell, which being in this case a cancer cell. So that's the main difference. This T cell targets virally infected cells and looks for the virus nucleic acid, whether it's an RNA or a DNA virus, whereas the CAR T cell targets and kills cells that have proteins that we select of choice on the surface. So CAR T cells have become the first living drug in medicine. We infused Emily in 2012 in April, and we just recently this summer got samples from her now 8 years later, and she still has functioning CAR T cells in here. It's a very complex process to make CAR T cells. The patient donates blood at a blood center, and then the cells are shipped to a manufacturing center, and approximately 10 days later, they are ready for use, but that involves the process that's done manually of inserting the lentiviral vector, activating the T cells, having them grow, so that about 1,000-fold during those 10 days. And then the cells are frozen and then infused in a simple intravenous, like a blood transfusion to the patient, which takes about 5 minutes. So then those cells stay on patrol in the patient, and that's what really makes them, what we call them, a living drug. So this is different from standard pharmacology and standard drugs that have been made over the years by the pharmaceutical and biotechnology industry, because CAR T cell is not a simple drug, but it's really a process, most akin to a bone marrow transplant. So the patient has the complex logistics I've described of donating blood cells that are manufactured and then reinfused and sometimes combined with other therapies. And at this point, these are autologous cells, meaning they come from the patient. And I'll describe some efforts now where we're working to make the cells be what are called off-the-shelf cells, with the CAR T cells made from other sources, such as cord blood or healthy blood donors or induced pluripotent stem cells. So Emily was treated in 2012 with a CAR that was made in my lab. It's usually said that cell and gene therapies take a very long time to get FDA approval, and in fact it did take a long time, but from the time she was actually treated, it was FDA approved and licensed in August of 2017 as the first cell and gene therapy approved by the US FDA. It's called Chimeric, and the first report in adults was 2011, and then in 2013 we reported Emily's case. And we treated many different cancers by now that are in the B-cell cancers that are leukemias and lymphomas that start in the bone marrow and lymph nodes, and they have all responded with varying degrees, leading to the FDA approval in 2017. So I want to harken back, when you know the cause of a therapy, you would think it would be pretty straightforward to make a cure for that. And this is, Cicero said this, that physicians consider when they've discovered the cause of a disease, they will have discovered the means of treating it. And, at the University of Pennsylvania, Peter Knoll discovered the first cause of cancer in 1963, which was the so-called Philadelphia chromosome, a disease that activated signaling and led to leukemia. And it took until the 1990s until Charles Sawyers, who you'll hear from later, found a cure by actually targeting the Philadelphia chromosome. So there's a very large gap in between the discovery of the cause of this particular form of cancer and the generation of a cure. In the case of cancer immunotherapy, it has been it's an old idea. Over 100 years ago, there were ideas of using the immune system to kill cancer, leading in the 1890s to William Coley, a surgeon who injected, he killed bacteria into humans who had cancer, and he sometimes had responses. But it took until 2011 until the first FDA-approved form of an immunotherapy was approved, the checkpoint therapies that led to the Nobel Prize that was awarded to Jim Allison and Professor Honjo in 2018. So a very long gap, and then in 2017 until CAR T cells were approved. So there are a number of lessons from this long journey. For more than a century, all these previous attempts had failed. That basically is due to the incomplete understanding of the very complex biology underlying the cause of cancer and how the immune system works. It required the innovations of recombinant DNA technology that allowed CAR T cells to be produced. And one implication, especially relevant to this meeting, is that means that healthcare funding, research and development policies need to have a long-term vision. You can't expect immediate miracles such as Operation Warp Speed, which is something I think we were probably discussing. So in this, we are having now a very large improvement in cancer survival, due to this long-term research in the United States and elsewhere, and seeing many different kinds of cancer, the survival rates going up, and this was reported by the American Cancer Society in January. And of course, soon thereafter, President Trump claimed credit for this lower cancer death rate, and just leading to it was due to many decades of research in the cause of cancer and to public health methods such as stopping cigarette smoking have led to major improvements in cancer survival. So back to some of the details of CAR T cells. The two that are FDA approved are Yescarta, which is marketed by Gilead, and Chimeric, which is marketed by Novartis. And there are many differences in how the details of how they are made, but they both have the same antibody that binds the CD19 molecule on leukemia cells called FMC63. They have, they're inserted in this case with a modified HIV lentivirus. This is a virus that comes from mouse called a gamma retrovirus. They have differences in how the CAR is designed, and then they have very long differences in how long they persist. The Gilead CAR in general disappears in patients after about 4 to 6 weeks, whereas the Chimeric CAR, now we have the first patient we ever infused was August of 2010, an elderly adult with leukemia. And we have found out this summer he still has CAR T cells more than 10 years later. So there is differential persistence. And I think in the long run, we want these to be physician controlled so that you can have cells that may have short-term survival or long-term survival, depending on that exact use that they have. And so there are many strategies now to enforce physician-controlled survival of these cells. So we're learning a lot now by studying the patients. So unlike other drugs, because our patients are genetically modified after they are treated, if you will, they're like a GMO. We can find those modified CARs because they have a different DNA sequence in them, or we can stain the surface of the CAR T cells, meaning they're normal circulating T cells, and compare the CAR T cells to cells that are not CAR T cells. And we have examples then of studying outliers where we've learned really interesting findings that have led us to develop more potent kinds of CAR T cells. And we've also had just blind luck, which I'll describe. So when Emily was treated, this is her fever curve here, with 2 peaks of basically converting from Fahrenheit to Fahrenheit over 106 degrees fever for 3 days. And she almost died from this. She had what we now call cytokine release syndrome. It's something we had never observed before after CAR T cell therapy. And she had multi-organ failure, was comatose, and in the intensive care unit. And it was only until we gave her a drug that's for rheumatology, arthritis, called tocilizumab, that she responded, and the fever went away. And the reason that happened was basically blind luck. And my daughter, Sarah, in 2001 got arthritis, and called juvenile rheumatoid arthritis. She's now a graduate student at Harvard, but still taking medicine. on a daily basis for her arthritis. In 2010, I was the president of the Clinical Immunology Society, and I had known that Tada Kishimoto in Japan had invented a drug that's now licensed to treat juvenile rheumatoid arthritis. And as the president of the Clinical Immunology Society, I gave him the presidential award for this notable achievement of a drug now that's widely used for many kinds of rheumatologic disorders and used worldwide. And then, when Emily we discovered she had very high levels of interleukin-6, which is what the drug Kishimoto had developed that blocks. We tried that for her, and it worked amazingly well. And it's now been FDA approved by the US FDA, and worldwide the use of tocilizumab, the drug made by Kishimoto, to treat the cytokine release syndrome of CAR T cells. So we can study CAR T cells in patients because as I mentioned, they're genetically modified. They can evolve in patients over time. We're required by the FDA to study the patients now for 15 years after infusion since they have become genetically modified. So this sets them aside from all other kinds of patients who are treated with other drugs and recombinant biologics. So there's an asymmetric form of monitoring to look for side effects in these patients. The pharmacology of these living drugs is very different from inert drugs that you administer and they just are metabolized away. We can follow the cells in these patients and they usually grow in patients until the cancer is eliminated, and then they go down to small levels of memory CAR T cells. And we found that these cells are very safe if they're autologous cells. We've never had them transform where the CAR T cell would turn from the CAR T cell into a leukemia cell itself. That is still a theoretic risk and may happen. And as I'll discuss, I think it's more of a risk for the so-called third-party CAR T cells that we and others are also trying to develop. So we're in this area now where the healthcare industry is changing. So we've had for many years a three-pillared system of global healthcare comprised of pharma, biotech, and then medical devices. And now we're transforming medical delivery to add a fourth pillar, which is cell and gene therapies. And they have different requirements than the ones we're mostly used to, and I'll discuss some of those now and later, I think, in our symposia. So one is that unlike other drugs, the autologous T cells are so-called N of 1. So they're manufactured on a lot-specific basis, and each one is FDA approved by testing and released. So it's more like a blood infusion where each one is specific and very different than the standard kinds of manufacturing that the pharma does, where they make one lot of a particular drug that may treat thousands and thousands of patients, and they use central manufacturing for that. So this is something that's a major issue under exploration in cancer therapy for CAR T cells. And some of the limitations of that are shown here, where I have described the complex logistics required to make cells from a patient. It's possible to do this, but it would certainly be desirable if we could use cells that are off the shelf from a separate donor. As you know, in red blood cells, you can have universal donors. If you have an O negative blood type, then you can donate blood to anyone. But we don't have that for T cells. Unless you have an identical twin, you have to have the T cells be derived from yourself for this kind of therapy to work. So it's not like it's not an easy problem to solve. There are other issues, which is right now the so-called vein-to-vein time, which is how long it takes to go from obtaining the cells to when they're infused in the patient. The initial time for this was 22 days when initial FDA approval occurred in 2017. we now have a manufacturing process at the University of Pennsylvania that's down to 3 days, so much shorter manufacturing time. Another issue is that there's a cell biology phenomenon called replicative senescence, initially talked about by Leonard Hayflick, where when cells divide, mammalian cells, their DNA gets shorter with each cell division. And this is a major issue with T cells in elderly patients, because if the telomeres get too short, the T cells arrest and are unable to continue to divide. So there is a glass ceiling, if you will, in treating some of the elderly patients with CAR T cells because their cells, if you will, are basically worn out. And this is an issue in general for an aging population. The immune system begins to flag, and even standard vaccines that work in all patients, all say healthy 30-year-olds, there's less than a 50% success rate with flu vaccines, standard flu vaccines, in people who are older than age 60. And in part, that's related to this Hayflick phenomenon. So I mentioned we study outliers, and because the cells can be retrieved from the patients. And shown here are the plots of the CAR T cells in the blood of patients for out to 2 years here. And you can see that there's a fairly uniform increase and decrease. But this one patient here in red had a delayed peak. So there's about 90 patients plotted here. But it turned out this one patient had the CAR T cell where the HIV lentivirus was used to insert the CAR DNA code. It landed in a specific gene called TET2. And then it made those cells be super CAR T cells. And we could show actually that this was actually the 10th patient we treated. That patient has been cured of his leukemia. And what we found out was that all the CAR T cells in that patient were the descendant of one super T cell. And so it taught us that just one CAR T cell can cure leukemia if it's good enough, which has profound implications on manufacturing. If we could manufacture many less cells and have the patient themselves be the bioreactor, it would be much cheaper and potentially more rapid to do this. One approach then that I mentioned is moving away from autologous cells and finding so-called off-the-shelf cells. And here I've compared some of the attributes of patient-specific cells that are N of 1 manufacturing versus off-the-shelf cells. And the cell source is obviously very different. They're called autologous if they're derived from the patient, and they're called allogeneic if they are derived from other sources. Obviously, this is with the autologous cells, you treat one patient at a time. And you could potentially treat many, maybe even 1,000 patients from each lot of universal off-the-shelf cells, which would mean that standard pharmacologic manufacturing used in a master cell bank could be used, and it would be much cheaper, and would then be in the shelf of pharmacies and ready for use. We know at this time, though, however, that the off-the-shelf CAR T cells have an unknown efficacy and persistence and safety profile, as opposed to the Chimeric, which we know can cure many patients with leukemia and lymphoma. There are a number of approaches to develop off-the-shelf CAR T cells. One that we're doing at the University of Pennsylvania. This is an ongoing project with Nikki Mason, who's a veterinarian at the University of Pennsylvania, where our approach is to use human T cell CAR T cells and use genome engineering to overcome the xenogenetic rejection mechanisms that would occur when the cells are infused from a human into a dog that has cancer, and then use that large animal model as a way to optimize off-the-shelf CAR T cells for patients. So we're actually using dogs with lymphomas in this trial and treating them with human T cells and comparing that to results where we treat with autologous dog CAR T cells. And so to do that, it requires genetic editing of the cells, and there are a number of ways now to do this, combining, say, technologies such as CRISPR-Cas9 to modify the DNA in T cells and improve them over what natural endogenous T cells would do. So we've recently reported a study where we use CRISPR-Cas9, which targets selected genes in the gene of, in this case, a T cell, and we chose 3 different genes to knock out using this technology that are on 3 different chromosomes, and then also to insert a T cell receptor in them. So we made and tested the ability of 4 different genetic modifications at once in the patient T cells in this trial using CRISPR, which was not even described until 2012. So it's a very recent advancement that we have the ability now to literally find a needle in the haystack of the 3 billion codons in human DNA to be able to target them precisely as we're at 3 different gene loci is to me an astonishing reality of the new technologies we have. And this was published, the first trial using CRISPR-Cas9 engineering in humans in February of this year. And we found it was safe in all 3 patients that we treated initially on this trial. So I mentioned we are in a process of moving from a 3-tiered healthcare delivery system to this 4th tier that includes cell therapies. And one of the issues in there is that the access is not uniform when one has a new form of technology, such as cell and gene therapy. And in general, when new technologies come out, they're much more expensive than mature technologies, and this is especially true of cell and gene therapies. So CAR T cells were, just developed by a handful of academic investigators in the United States, and then initially approved in the United States. The first trials began in 2006, and now if you look at the clinicaltrials.gov site, you can find that there are now more than 600 CAR T cell trials ongoing in the world. And most of these trials actually now are in China. It's amazing how fast they have moved into this field. In general, China has been thought of in their pharmaceutical industry as an industry that generally made knockoffs and Me Too drugs and was not thought of to be high in innovation. But we're seeing a lot of innovation in China now, and a real challenge to the dominance of U.S. Academic development of new biotechnologies. And so I think this is a really important observation. In general, you can see that this is not just that Asians are more gung-ho to develop and test new kinds of trials with CAR T cells, because in Japan, it is so neighboring close. There's only 11 trials open compared to the 300 in China. So the Chinese society is very aggressive to adopt new technologies, and the government has promoted policies to invest in this, and this is something that we need to consider. The Wall Street Journal and others have talked about the so-called $1 million cancer treatment, who will pay, and this is an issue with cell and gene therapies. And it's really a looming issue because in the case of acute leukemia that we got the first FDA approval for, there's only 5,000 cases a year in the United States, but it's thought that the most common blood cancer in adults is myeloma, where there's 30,000 cases approved or diagnosed every year, and it's thought that CAR T cells will be approved by several companies later this year or early next year in 2021. And myeloma is an awful disease. In general, it's not curable. And, patients live 8 to 10 years after diagnosis. And the cost of their treatment is about $10,000 to $12,000 a month, and leading to a huge financial cost, so-called financial toxicity, to treat that disease. It's only 2% of all cancer in the United States, but it accounts for about 7% of all costs that the U.S. pays every year for cancer care. So there are many ways to make CAR T cells cheaper. One is manufacturing technology. A second is using the so-called third-party CAR T cells that I described, and also to use cheaper manufacturing labor costs, which is, for instance, offshoring this to areas where there is cheaper manufacturing. And I'm currently exploring that in both Costa Rica and in India, where the cost of manufacturing is much less. So CAR T cells are made, right now at this point by scientists on a one-to-one basis. Automobiles, were initially made on a one-to-one basis on, the Henry Ford, assembly line. And now most cars are made by robotics and assembled without human hands. And that's what needs to happen with CAR T cells. Our CAR T cells are shown here, our manufacturing process that was developed in my lab in 1995. This is a microscopic image of T cells bound to brown spherical 5-micron polystyrene beads that have antibodies that we covalently attach that activate the T cells so that they proliferate. And these are also paramagnetic, and so we remove the beads at the end of culture using magnetic fields as shown here. And then we have pure T cells later, which are infused and look sort of like milk chocolate here. So that's a manufacturing process that's used by Novartis, but it's very labor intensive. It requires highly trained scientists and technicians to do this, and we need to develop automated systems. And there's a lot of effort now in the biotechnology industry to develop these advanced manufacturing, and using actually many of the lessons from Detroit, where engineers have shown how cost follows different scale outs of the number of patients that are treated. And if you make a Lamborghini, those are in general made by hand, and they're very expensive because they're put together by highly trained mechanics and technicians. Whereas an automated Volkswagen that's put together by robotics is cheaper by more than a log scale. So there are many different groups now brought together to learn how to make cheaper cell and gene therapy. And there's ideas that this is an industry that's different than the standard pharma industry. Cell and gene therapy, because it begins where the patients are at hospitals, is somewhat more dependent on the development of new therapies with the academic institutions. And the industry now is working with many different groups to make this logistically complex process simple. And and there may be one approach. There's a paper published by Beth Schachter in Nature Biotechnology proposing that we could accelerate the development of more advanced cell and gene therapies by having government, industry, and philanthropic partnerships. The UK has this, where they have made central manufacturing plants, which are the expensive rate-limiting step to make new cell and gene therapies. And they have made those through governmental forms, and then new academics and fledgling biotechs can use that to test initial trials, concepts, de-risking them and decreasing the total cost. This may be a way to accelerate the development of cheaper and more affordable cell and gene therapies. One concept that's approved or emerged is shown here, which is that we may hit peak serum. And, serum is used in the culture of most cells, and it's derived from cows. And there are so many cows that can be donors for calf serum. And this paper published in 2012 shows that the peak serum levels, that one blockbuster cell therapy could overwhelm the supply of the world's supply of cows to supply serum. And they say potentially that the concepts of peak oil and peak water have spurred innovation and not disaster. And hopefully that the same may be true for peak serum. So there's a lot of effort now to make cell culture conditions that are independent of the requirement for calf serum. There are economic forecasts showing that the cost for cell and gene therapy is related to the number of patients that are treated. So for very rare diseases, the costs have been over $1 million per patient. And now for the cell and gene therapy, where for a lymphoma, it's down to $375,000. And it will come down, I think, by an order of magnitude in the next 5 years. But this has huge implications for the Center for Medicaid, CMMS, which sets the price of drugs in the United States. There have been now a number, actually 500 different companies start. When we published our first paper about Emily in 2012, there were no CAR T-cell companies, and now there are 2 marketed in the US. But there are hundreds around the world now, as shown in this dot plot of companies testing various ideas. So it's a very active new area of the biopharma industry. So there are many aspects of this that are specific to cell and gene therapies and that I've highlighted throughout my presentation. And I think we can discuss some of these later today in the panels. So in summary, there are many kinds of CAR T cells in development now. It's a new pillar for the medical therapy in the US and worldwide. Cost and scale are the major problems facing the field. In genome engineering and cell omics at the DNA, RNA, and protein level have a major potential to synergize to make more potent next generation. I'd like to thank you for your attention, and I look forward to the panel discussions.
So let's see. I said that you could participate in asking questions by going to a website, and I neglected to tell you what that website was before. If you have a question for Dr. June, go to pollev.com. That's POLLEV.com/Nobel 56. Or if you'd like to participate by text, go to 22333 and text Nobel 56. Since we can't all be on campus for this Nobel Conference, we decided we would bring you some images from the campus to make you feel like you're really there. For those of you who have great familiarity with the Gustavus campus, you will say, where is Lisa standing in front of? And the truth is, we've made a pretend room that doesn't really exist, but that is about where North Hall would be. And so just imagine, if you will, a viewing platform somewhere out where North Hall is, looking over the Old Main, my building. So at any rate, my colleague, Professor Laura Burek of the Biology Department, is in a real room. In fact, one of the exciting real new laboratories at Nobel Hall. So welcome, Laura, to this virtual conversation.
Thanks, Lisa.
Is this your lab by any chance?
It is not my lab. My lab is just down the hall and around the corner, but
Wonderful.
It's pretty close.
Excellent. And I'm certain that you've been enjoying settling into the new digs.
Yes, they are beautiful. It's sort of amazing to see what a transformation has been made. I have so much more space and collaboration room and all sorts of things. It's a great building.
Wonderful. Wonderful. So I, as you know, describe myself as the chief learner of Nobel Conference, and so I have the delight today of getting to talk with you a little bit about Dr. June's talk, because it turns out you know a lot more about what he was talking about than I do. So one of the things that I'm really interested in is this difference he talked about between viral editing and CRISPR gene editing. So what's the difference between those two, and why would you use one rather than the other?
Yeah, so both of them are ways to actually modify the genome of a cell. The difference comes in the way that they do it. So the HIV vector that he mentioned is sort of part of a large family of retroviral vectors. And so these take advantage of the fact that those viruses, when they infect us, part of their genome actually inserts into our DNA. And so that insertion into our DNA is what allows the virus to have a long-term infection, but it also then essentially changes our DNA. And so by taking away the infectiousness, but leaving the insertion, you can pretty much put in whatever you want. And so that's a technology that has been around for quite a while, and it's really, really good at putting in pieces of DNA. What CRISPR does that's a little bit different is it actually allows you to target where in the genome you want to make a change. And so if you want to change something about the existing DNA, CRISPR is the way to go. So the retrovirus is really good for putting something in, and the CRISPR is really good for changing the DNA. And so the CAR T therapy, the way it's been used in clinical trials, really what they need to do is insert that CAR into the DNA. And so therefore, the retroviral vectors work great. For some other types of uses of something like CAR T therapy, for example, to treat solid tumors, they need to make more changes. And so therefore, they've used something like CRISPR, which can actually change the DNA to be able to make those types of changes.
I'm sorry, go ahead.
Oh, I was just going to say they're both changes in the genome, but it's a little bit more like, do you want to put something in, or do you want to change what's already there?
I noticed you're not saying take something out, which is my sort of layperson's understanding of how CRISPR works. They always depict it with a scissors, and I always think, oh, you're just taking something out. Not so.
So really what CRISPR does is it makes a cut at exactly the right place.
Okay. And there
so
you can stick something in if you need
don't
to.
give it anything to put in, it will actually make a tiny little deletion, because the process of fixing that cut will make a little change. And then, yes, you could delete a gene.
Okay.
But you can also put in a new piece of DNA, and then that cut will be exactly where the new piece comes in, and you can really just sort of fix what's there.
Okay. I'm going to interrupt our discussion just to invite the audience to participate in a little poll that we created about Dr. June's talk. So again, go to that same place, pollev, that's pollev.com/Nobel56, or again, the phone number is 22333, and text Nobel56. And we will, and you can see the poll on your webpage when you enter that address. And while the poll results start to come in, I have another question for Laura. Was there something particular about the HIV virus that made it particularly optimal for this particular kind of insertion? Do you happen to know?
Yeah, so I think it's a combination of the fact that HIV has been very well studied, and so the integration process has been very well studied. That I think might be one reason why it's been used so much.
And indeed, Dr. June studied HIV,
yeah,
I believe.
Definitely. And I think that the fact that we know so much about its pathogenesis as well allows us to be very certain that we are getting rid of everything that is pathogenic if you were to use that type of vector. So those are just a couple of the reasons.
Brilliant reasons. Interesting. Now, of course, as a dog person. My ears pricked up when he started to talk about the ways in which they are conducting trials using dogs. And indeed, my web feed all year has been sending me things about dog cancer as I have been collecting up cancer information for the Nobel page. So in the case of trials they're conducting with dogs, those cells are normally rejected by the body. Am I understanding that correctly? Why does that happen?
Yeah, so every person has specific markers on the surface of your cell.
Okay.
And your immune system is trained during the development process to be sort of ignoring of those markers. So when your immune system develops, all the T cells and B cells are actually generated through this random process where they put together all sorts of little pieces to essentially look for any possible thing out there.
Okay.
But during development, they're trained. So essentially, if they recognize you, they're killed. And so anything that's not you could potentially be recognized by the immune system. And so therefore, the dog's immune system essentially hasn't been trained to ignore human cells. And so therefore, we have to trick the cells so that they now don't have any of those sort of surface markers that would make them sort of be seen by the dog's immune system and therefore be rejected. And so like when you think about organ transplant, you think about how there has to be a very specific match that occurs between the organ that's being put in and the patient. It's that same idea that sort of allows for organs to not be rejected, although in those cases, they do have to take immunosuppressants so that they're not rejected. And so when he's talking about this idea of allogenic T cells, essentially that's the problem that has to be overcome. They have to figure out how to make it so that the T cells from someone else don't recognize anything about other people. They are sort of trained specifically for one person, and so then we have to train them to sort of be ignoring of lots and lots of people instead of just the one person.
And he mentioned, of course, universal donor in with respect to blood, and there's sort of no universal T cell. And of course, I guess when you think about it, what good would an immune cell be if it was just universal, right?
Yeah, so if it was universal, then you could maybe fight one infection, but you would only be able to, let's say you had universal influenza cells, you could potentially fight influenza really well, but any other infectious disease, you would be completely susceptible. And so it's the randomness that allows the immune system to work so well, but it's also the randomness that makes it so hard to sort of use someone else's cells in this type of immunotherapy.
There you go. There you go. Well, it looks like we've gotten some results in from our poll. Which statement best describes your thoughts on immunotherapy? And Laura, I'm not sure you can see these results, but
I can.
You can. Oh, well, wonderful. So it looks like a majority of folks say, as I certainly would have until now, I didn't understand immunotherapy could lead to long-term cancer cure, but a substantial, 14% of people say, I worry about those negative long-term consequences of modifying an immune system. What do you notice about those results, Laura? Does that surprise you at all, particularly the concern?
No, I think it makes sense. So when we think about immunotherapy, they are incredibly powerful treatments.
Okay.
And so essentially that power is what makes them so effective, but that power is also what makes them potentially a little scary. So if we think about all sorts of different diseases that are related to autoimmune diseases, those are all something that is potentially a risk of some of these immunotherapies. And so the trick is to make sure that that's not happening. So most people that are treated with immunotherapy today, including the patients that undergo CAR T therapy, are generally quite sick, such that without the immunotherapy, they might not have very long to survive. And so if you're sort of weighing the pros and cons, if you're thinking about the possibility of a long-term effect 15 years down the road
Right.
versus a prognosis of a year, I think that that's a pretty easy choice to make.
Right.
If you're thinking about cases where patients might have a prognosis of 20 years, so a lot of prostate cancers, for example, are very slow growing. And so if you have a very slow growing prostate cancer, I think it becomes a much more difficult decision to think about which of those is the greater risk. And so I think that personally, immunotherapy is incredibly promising and has incredible benefits for a lot of situations, but we should be sort of thoughtful about when to weld the power.
Well put. I'm aware that we have a hard stop at 11:45 so that our audience can go to discussions, and that Zoom code should be available to you on the webpage. But I think we can squeak in one more question, and I think it might be as a wild card question, but it's, okay, tell us about cow serum. Why can't we just use, I don't know, something else?
Yeah, so he mentioned the idea of peak serum in his talk, and some of the audience might be wondering why do we need cow serum? And so essentially it's because human cells are designed to be in a social setting. So it's called actually social control over the growth. So they're designed to be part of a multicellular organism. And so therefore, in order to replicate, even if they're potentially cancer cells, they still need all of these growth signals in order to replicate. And so if you want to grow human cells in a dish, you need all of these signaling molecules, all these different growth factors. But it doesn't really work if you just have one. You need a whole slew of them. And so you could theoretically make each one of those individually, but that might also be tricky. And so the easiest way to do it is to use serum from a cow, because cows are large, relatively speaking, and so you can get a large number of volume out of a given animal, as well as the fact that usually it's actually fetal bovine serum. So when you think about development, early in development, there's all sorts of growth that happens. And so essentially it's the fetal bovine serum is used because newborn developing cows have lots of cell growth, and so they have lots of growth factors which make the cell culture work.
Neat.
So
That's fascinating. And we are just about at time to go to audience discussions. Audience, I remind you that you can either join a Zoom discussion led by one of my colleagues, or you can sit back and relax and watch pre-recorded content created especially for today's conference. So thanks again, Professor Laura Burek of our biology department, and thanks for all that you've done for the conference.
thanks, Lisa.
See you later. So audience members, please feel free to join a Zoom discussion or watch that stream of interesting videos. Let's see, this afternoon, what can I tell you? We'll start with a healthy cooking demonstration, a poetry reading, and then a segment on yoga for cancer, followed by a tour of art that's happening in the Schaefer Art Gallery.